Hello and welcome to Bio40 Pre Lecture 12. Today we're going to talk about the ways we've hijacked the process of DNA replication for our own purposes. The ability to synthesize DNA in vitro, that is in a test tube as opposed to inside a living organism, has enabled some of the most important techniques used in biology research. In vitro replication greatly simplifies the whole process. We of course still need a DNA template to work from. Instead of using a primase to make an RNA primer, we use short DNA chains called oligonucleotides, and would still also get referred to as primers. The primers are necessary because we're still using DNA polymerase, so we still have to provide a 3' hydroxyl for it to work. We also have to provide the substrates, the dNTP building blocks. And what about all the other proteins? Well, we actually don't need them anymore. They can be replaced with the right temperature manipulations. The first step in vitro replication is denaturation. The DNA is heated to around 95 degrees Celsius, just below boiling. At this temperature, the hydrogen bonds holding the double helix together break and the strands separate. This is called denaturing. With this step, we no longer need an initiator protein, helicase, or topoisomerase. Next, we cool the DNA, which allows the primers to base pair with the template. Our DNA primers remove the need for primase. And since they're DNA and not RNA, we don't need a second polymerase with 3' to 5' exonuclease activity to remove and replace the primers. And this means there are no nicks that require a DNA ligase. All we need to do is add polymerase to extend the DNA chain from the end of the primer. There are two major technologies that rely on this process. The first one we're going to discuss is sequencing, which allows us to determine the base sequence of an unknown piece of DNA. In sequencing reactions, we add a special substrate, a dideoxynucleoside triphosphate, or ddNTP. ddNTPs differ from dNTPs by the chemical groups on the 3' carbon. ddNTPs lack the hydroxyl there. And by now, I've hopefully given you the impression that the 3' hydroxyl is pretty important for replication. A ddNTP can still be incorporated into a DNA chain by DNA polymerase, but the lack of a 3' hydroxyl means the chain can't be extended any further. This was used in the earliest form of sequencing, called Sanger sequencing. Named for Fred Sanger, this is a process that earned him one of his Nobel Prizes. With this approach, you would set up 4 different reactions. Each would have the same template DNA, primer, polymerase, and dNTPs. And one of the dNTPs would be radiolabeled, since the radioactivity would be a useful way of visualizing the newly synthesized strands. Then each reaction would have a small amount of one type of ddNTP. One would have ddATP, one would have ddCTP, one ddGTP, and the last would have ddTTP. One problem with our approach is that we need to know the sequence of part of our template DNA, which is an issue since the whole point of the process is to determine the sequence of an unknown piece of DNA. The reason for this is that we need to make sure we have a primer that always binds to the same part of the DNA template. But we only need to know a short region, usually around 20 base pairs for the primer to pair with. And we have ways of attaching known pieces of DNA to the ends of our mystery pieces when we need to. Now we go through our in vitro replication process. We heat the template to denature, cool to let the primer anneal, and use a polymerase to extend the chain. But in this scenario, we have the ddNTPs present, which can terminate the process. Let's say we're looking at the reaction that has ddATP in it. So the first time the polymerase gets to a T in the template, it could add a dATP or a ddATP. If the ddATP is added, there is no 3' hydroxyl and the elongation ends. But if a dATP is added, the process can continue until the next T in the template, where again the chain could be extended with a dATP or terminated with a ddATP, and so on. So in the end we have a bunch of different products. We'll do one final denaturation step so that we can focus on the new chains, which we can clearly see have several different lengths. In this case, you'll get a chain that is the length of the primer plus 3 bases, another that's the length of the primer plus 7, primer plus 9, plus 10, and plus 15. This tells us that the 3rd, 7th, 9th, 10th, and 15 bases after the end of the primer are A's, and the other 3 reactions will tell us which positions are C's, G's, and T's. Of course, we can't actually see these lengths just looking at the reaction tubes. To see them, we use gel electrophoresis. The 4 different reactions are added to 4 different wells at the top of an acrylamide gel. Then a current is applied that pulls the DNA toward the bottom. But the gel matrix greatly slows down big pieces of DNA while letting smaller ones pass relatively unimpeded. In the end, the DNA is separated by size, with the smallest pieces at the bottom and the largest at the top. We still can't see the DNA here, but if we hold a piece of film to the gel, the radiolabeled DNA will expose the film in the places the DNA is present. As a result, we see a banding pattern like what is shown here. The exposed film is called an autoradiograph. Now we can use a banding pattern to determine the sequence. Here, the smallest band is in the lane with the reaction that had DD-CTP. The chain was terminated with a C, so C is the first base past the primer. The second smallest band is in the T lane, so T is the second base. Then A, G, G again, and so on. We can keep reading the sequence moving up the gel. Note that this is a sequence of the newly synthesized strand and not the template. While the original form of Sanger sequencing will get the job done, it had several issues. You need 4 reactions just to get one sequence. The process requires using radioactive elements, which is a huge safety issue. The gels are quite large and can be very hard to make and use. And it just can't sequence very much at once, maybe just a couple hundred nucleotides. To go any farther, you need to use the sequencing information from the first round of reactions, make a new primer that will pair with the end of your newly found region, and set up 4 more reactions to get the next couple hundred bases. Luckily, there's a modern version of this sequencing approach that solves many of these issues. In this method, you only need to set up a single reaction. You still have the template, primer, polymerase, and DNTPs, but this time none are radiolabeled. Instead, we're adding all 4 DDNTPs together, and each type of DDNTP has a fluorescent label with a different color. We can do in vitro synthesis just like before, except now a DDNTP could stop the reaction at any position. Next, instead of running the reaction on a gel, we put it through a very thin capillary gel tube. Using electrophoresis, this gel tube does the same job of separating the newly synthesized DNA by size. The smallest pieces move through the fastest. At the end of the tube is a laser that excites the fluorescent molecules, and a detector that tells you what color of fluorescence just passed through. In this case, the detector first sees blue fluorescence, which is the color the DD-CTP was labeled with. So the smallest piece of DNA was terminated by a DD-CTP, and our first base must be a C. Next, the detector will see red, which is T, then green, which is A, and so on and so forth. This information is typically displayed in what we call a chromatogram, shown here. The fluorescently labeled DNA is detected as these series of peaks. You can see this one begins green peak, 2 blue peaks, red peak, black peak, for A, CC, TG, and so on. This modern variation can usually sequence about 1,000 bases. Better than the previous method, but still very small for some applications. This certainly won't work very well for people who want to sequence entire genomes with millions or even billions of bases. For that, people use what we call next-gen or high-throughput sequencing, which you can learn about in some upper division courses. Now let's talk about the second major application of in vitro synthesis, polymerase chain reaction, or PCR. This process is used to greatly amplify tiny amounts of DNA, and it allows you to amplify a very specific region within the DNA. It has applications ranging from lab work to medical tests to forensic investigations. Scientist Kary Mullis invented it in 1983 while working for the Cetus Corporation. He reasoned that with multiple primers and the right use of heating and cooling cycles, he could amplify DNA with a polymerase and no other enzymes. Coming up with this method earned him $300 million when he sold the patent, and a Nobel Prize in 1993. This is somewhat unusual, since Nobel Prizes are rarely awarded to people who invented technology, but did not play a role in discovering the underlying scientific principles. But there's no denying how influential PCR has been. So just like before, we have a reaction with a template, DNTPs, and polymerase. No DDNTPs this time because we're not trying to terminate the extension early. And instead of 1 primer, we have 2. Let's say we're trying to amplify this specific portion of DNA. We get primers that base pair with both the top and bottom strands, and we make sure they're positioned in such a way that extension from the 3' end will cover the region we're trying to amplify. Again, we must know something about the sequence of the DNA to get the right primers. Now we proceed with the normal in vitro replication plan. Heat to denature the DNA, cool to let the primers anneal, then extend the chain with the polymerase. This creates 2 of what we call long strands. They begin where our target sequence begins, but extend past the end of the target. Then we go through the whole process again. Denature the DNA, anneal the primers, and extend. The primers that anneal to the template DNA will give us 2 more long strands. But primers that anneal to the long strand of the previous round can only go as far as where the other primer began. The resulting piece of DNA only contains the target sequence. And every time we go through this process, we double the amount of target sequence we have. Let's look at it this way. We start with this one piece of DNA and want to amplify the bracketed region. So we design primers that anneal at the right positions, and do 1 round of replication that gives us 2 long strands. Denature, anneal, and extend again, we get 2 more long strands for a total of 4, and then 2 target sequence strands. The next round adds another 2 long strands, but 6 target. At 4 rounds, you can see that the target sequence is the predominant form of DNA. And after 30 cycles, we still have just our original 2 parent strands, 60 long strands, and over 2 billion strands of the target sequence, or 1 billion pieces of double-stranded DNA if we don't do a final denaturation step. For a short target sequence, each cycle only takes a few minutes, so this whole thing can be done in a matter of hours. The earliest iterations of PCR had some big problems. It was first done using a polymerase from E. coli, which works best at human body temperature, or 37 degrees Celsius. And what do you suppose happens to the protein when the temperature is raised to 95 degrees for the next denaturation? If you guessed it's irreparably damaged, congratulations. So new polymerase needed to be added every cycle, which is both expensive and time-consuming. But there are some organisms that are adapted for higher temperatures. The bacterium Thermus aquaticus, abbreviated TAC, lives in hot springs. Its polymerase works optimally around 70 degrees Celsius and can tolerate short times at 95 degrees. It remains stable throughout the whole process. This gel shows PCR with E. coli polymerase on the left and TAC on the right, and it illustrates how much cleaner the results are with the TAC polymerase. The other major advance for PCR was automated machinery. At first, scientists needed to manually move the reaction tubes between the denaturing, annealing, and extension temperatures, which meant babysitting the reactions for hours. The earliest PCR machines had different heat blocks that maintained the right temperature for each step, and a mechanical arm moved the tubes between the different temperatures at the proper intervals. These days, things are even better. We have thermal cyclers that have just one heat block which can be rapidly heated and cooled to the proper temperatures. We also have polymerases with greater fidelity. The polymerase from the Archaea species, Pyrococcus furiosus, or PFU, is also stable at very high temperatures, but unlike TAC polymerase, it has a proofreading function and makes significantly less errors in replication. And with that, we're through with replication. But speaking of errors, next time we start discussing ways in which mutations can be introduced to DNA.